Assalamu alaikum and a very good morning to my students of Green Valley Schools, Metric and CBSE. Today I am going to continue with this replication of viruses. In my last video we have seen the step 1 replication of virus, that is adsorption. In other words, we can say attachment of virus to the cell membrane of the host. Today we are going to continue with the second step, that is penetration. Penetration is nothing but entry of virus into the host cell membrane. Enveloped viruses and non-enveloped viruses. Enveloped is nothing but an extra coating, whereas non-enveloped viruses are naked viruses. Non-enveloped viruses usually they enter into the host membrane directly by penetrating the DNA. Once it is attached, once the virus gets attached, directly the DNA or genetic material is inserted inside the host cell membrane. That is one of these steps of penetration. Whereas enveloped viruses, they also can directly penetrate the DNA or RNA inside the host cell membrane. Apart from that, the viruses with the capsid or envelope gets engulfed by the host membrane through a process called as endocytosis. Can you see this first picture where the cell membrane has engulfed the virus directly inside the host membrane? This is nothing but the step 2 of penetration. Then we have step 3, that is uncoating of genetic material. Means to say, the genetic material virions, that is, it is also known as DNA or RNA, it is surrounded by a protective layer of capsid. The capsid dissolves and the release of DNA or RNA takes place. This is the third step called as uncoating. Usually removal or degradation of capsid, thereby releasing the genome or genetic material inside the host cell, is uncoating. The viral genome is transported to the site where transcription or replication takes place. Once the virus is released into the host membrane, the genetic material of that virus goes where transcription takes place, actually host transcription takes place. It interferes in the transcription process and it starts replicating itself. Virus starts replicating itself. They multiply by themselves by interfering into the host transcription process. In some viruses there is no degradation of capsids. This capsid protein itself plays a vital role in transcription and replication. Can you see the third here, second diagram? This is nothing but uncoating. Can you see this? A virus is uncoated and release of genetic material starts, that is DNA or RNA. So let me revise, students, for 1 minute for you. Penetration is nothing but entry of viruses or entry of their genetic material DNA or RNA inside the host cell membrane, either directly or through endocytosis. Then we have uncoating of capsid takes place or proteins takes place. For few viruses, capsid will not dissolve. Capsid itself plays a vital role in transcription and replication of viruses. Whereas usually capsid dissolves, the DNA or RNA is released into the host membrane, they go and interrupt the transcription process of the host. They use the transcription process for themselves and starts replicating. In my next video, we are going to continue with replication of viruses. I request the students to please see the video, revise. If you have any kinds of doubt in the chapter of viruses, please, all of you know my number, please send me a message. I'll clear your doubts. Stay safe, stay at home. Thank you very much.